Hello. We appreciate the opportunity to present our work entitled Eclecticism in Cladistics, Convergence among Opposing Optimality Criteria May be a Hoax. We divided our presentation into three parts: a short introduction, three questions, and our conclusions. We will present methods and results for each question. Rindel and Brouwer, 2011, heavily influenced our work. Almost a decade ago, the authors reviewed 1,000 papers published on molecular phylogenetics and evolution. 504 of those articles used opposing optimality criteria for tree reconstruction. Only 5 papers shown trees that strongly disagree with each other. The authors concluded that either most actual datasets converge when analyzed under different optimality criteria, or there is a bias in the publication of phylogenetic analysis. This bias could be due to a false notion that when different methods support the same topology, this somehow lends extra support through their phylogenetic hypothesis. It is also possible that authors feel socially compelled to methodological syncretism, since it is a pervasive practice. 3 questions could help us address this problem. The first one: are phylogeneticists frequently publishing convergent trees? If so, should we expect trees from opposing principles to converge repeatedly? Moreover, should we expect convergency among false discovery rates to be negatively correlated? To answer the first question on the pervasiveness of comparing opposing phylogenetic criteria, we reviewed 545 articles of molecular phylogenetic and evolution. We evaluated every paper that included phylogenetics. All sources of data were accepted, no matter if it was molecular, morphological, or otherwise. We also looked into every article, no matter if the authors used parsimony or not. And the results show that 311 of the 545 papers used multiple methods, and 295 of those, approximately 95%, reported convergence among opposing criteria. To answer the second question on how often trees from different methods should converge, or how often we should expect them to converge, we use computer simulations. We randomly selected 154 datasets from 3 bays and used each one of them to simulate alignments with different techniques. Alignment lengths was proportional to the number of terminals. We performed simulations so that the generated matrices would have much higher retention index than the original. We repeated simulations 100 times per dataset, generating 15,400 concatenated matrices. We analyzed each one under parsimony, maximum likelihood, and posterior probability. The total amount of 3 search experiments was 46,200. We used normalized match split distances to calculate the distance among trees, and we considered that trees converged if they were less than 10% distant from each other. We observed convergency in some replicates based on 54 of 154 datasets. Only 1,204 of 15,400 triplets of 3 search experiments, which is about 80% of the total, shown here in a darker shade of gray, were convergent. The difference between the distance from selected and rejected trees to the original tree was only statistically significant in 7% of the 154 datasets, about 5%. When the distance from selected and rejected trees to the original trees was statistically significant, it was impossible to predict which group was better. For example, take these 2 examples. If we only accept trees when the 3 criteria converge, the trees we would accept this way would be closer to the original tree than the rejected trees on the dataset on the left. However, at least in some cases, such as on the dataset on the right, the selected trees would be worse than the rejected ones. In most cases, though, the median was the same, and also it does not seem that comparing methods help to reject the worst trees on the top of the graphic. Finally, we could not produce enough data to answer whether comparing two criteria is better or worse than comparing three criteria, yet it seems to be a tendency for the rejected trees to get better the more methods we are comparing. Finally, to answer the last question, should we expect convergency and false discovery rate to be negatively correlated? And here we want to introduce a mind experiment we call the three archers. Imagine, if you will, three archers that are equally good. They are all excellent archers, and they will shoot their arrows towards the same target in 2 consecutive rounds. Let's imagine they cannot affect each other in any way. Now imagine that you are betting on the green archer, but you cannot see the target. You are only told how far from each other the arrows have landed. If all the archers hit the same spot in the first round, but hit different places in the second round, when the green archer hit the target, we show you the 4 options in this table, but before you make a choice, let's talk a bit about Bayes' theorem. Bayes' theorem helps us calculating the probability of a hypothesis given the evidence. Let us say, for example, that you are part of a population of 1 million people. About 1% of the population is affected by a certain condition, that is, your prior. We have a test with 99% sensitivity and specificity, which means that the analysis identifies 99% of the people with the disease and misdiagnoses only 1% of the patients. In this scenario, if we test everyone, we should expect 990 true positives and 999 false positives. That is 9,990 false positives among all 10,980 positive results. So the chance of you getting a positive on the test and have a disease is about 90%, and the rate of false discoveries is 91%. These values can be calculated using Bayes' theorem that states that the probability of the hypothesis given the evidence is equal to the probability of the evidence given the hypothesis, which is also known as the likelihood of the hypothesis. The likelihood of the hypothesis is multiplied by the probability of the hypothesis, which is the prior, and then divided by the posterior probability of the evidence. To make it clearer, this is the calculation of the same 9% posterior probability we discussed 2 slides ago. What if we replicate? As you may know, if we replicate the test, we update the prior, which results in a much higher posterior probability than the first test. So if you get a positive result, replicate the test, and get another positive result, the chance of you having the disease is about 91%. But if we do not replicate, but instead we conduct several independent tests and decide to accept a positive result only if every test is positive, well, in that case, instead of replicating the test, we would only be perform independent evaluations. We should only then multiply the posterior probability of each test, as we show here, which would always decrease the final posterior probability and increase the false discovery rate. It is easy to demonstrate that. In here, we use computer simulations to simulate the very same scenario of the population with 1 million people, 1% of which affected by a disease. We compared, using 1,000 repetitions, the rate of false discovery if we only accepted a positive result in all independent tests. Also, if all independent tests also gave a positive result. All the tests were the same, and we just set the sensitivity and specificity of the test to different values as we show here. As you can see, the rate of false discoveries increases the more tests you compare. If we only accept the result when multiple independent methods converge to it, the false discovery rate should increase the more methods we are comparing. Now let's go back to our mind experiment. Knowing all this, how would we bet on the green archer if we are told he hit the same spot twice? Well, in this situation, is similar to replicating the test and getting the same result. And since we are told that the archers are very good, it is likely that he hit the bullseye both times. But this decision is much more based on the replication, 2 arrows in the same spot, than on the position of the other arrows. And what if we are told that the green archer hit the bullseye only once? Now, knowing that the more independent tests we compare, the higher the false discovery rate, perhaps we should choose option number 2. In conclusion, convergency is rare, and if it's difficult to observe it in computer simulations with controlled amounts of homoplasy, perhaps they are even more rare using real data. This means that parsimony, maximum likelihood, and posterior probability don't seem to be redundant, and that frequent convergency among methods in phylogenetics may be due a bias caused by, in part at least, false assumption regarding what convergency represents. It is important to note that results of one method are not necessarily a corroboration of other methods, even when the results are the same. Finally, we reiterate that what we consider the most important claim of Rindel and Blauer 2011. Researchers have to select criteria according to their philosophical thought, to their belief, and they cannot avoid defending one method over the other, even if different methods result in the same hypothesis. We thank you all. This is our emails. If you photograph this QR code, it will take you to a website containing this presentation, including bioinformatic pipelines and other supplementary material. Thank you.